As we continue looking at mitosis, we've already looked at prophase. That was the first phase of mitosis. So today in this video we're going to look at the other three. Remember, the phases of mitosis form the PMAT. Prophase we've already done, so we're going to be looking at M, A, and T in this video. Metaphase is what the M stands for. This is the second phase of mitosis. So again, this is all about the nucleus and the chromosomes in the nucleus dividing. So we have the chromosomes are going to line up down the center of the cell. That's what the meta is referring to, is the middle of your cell. And the microtubules are going to connect to the centromere. We already saw that in the prophase picture. They've already started to connect to the centromere, but now they're going to be connected on both sides. So that you're going to have a spindle on the right and left side. So it's going to look like this. So this is metaphase, and you do need to draw this. You also need to label it. So again, if you don't know what these are, you have labeled these already, so you don't have to relabel it. But again, we have the centrioles. We have the spindles. Notice that the spindles are fully developed now. In prophase, they were only maybe partially across. Now they go from one centriole clear to the other. We have our chromosome. And the chromosome has, again, the two sister chromatids. They're identical, and it has the centromere. So notice that there's a spindle on each side of the centromere holding it all together. So feel free to pause that if you need to continue drawing. This one you don't need to draw. I kind of like seeing multiple ways that something can look. So this one shows you kind of a metaphase plate. So again, it's just that middle portion of the cell. You can see the chromosomes are lined up right along the middle, and the spindles still are connected to both sides. And there's extra spindles in there as well. So that's just another metaphase drawing. Now if we look at the third phase of PMAT, the A, that is going to stand for anaphase. And anaphase is the third part to mitosis. And there's a few things going on here. First of all, the centromere, and again the centromere is the part of the chromosome right here that holds your sister chromatid to your other sister chromatid. The centromere is going to split in half. And because it's not holding the two chromatids together anymore, those chromatids are going to get pulled in opposite directions, and they're going to get pulled by the spindles toward the centrioles. It says that they're going to be near the poles. So the poles would be to the clear opposite ends of each other where the centrioles are. And then lastly, anaphase is over when the chromosomes stop moving. So from the time the chromosome gets pulled apart and it's in the middle and it's going to get pulled all the way to the time that this half is all the way over here and this half gets pulled all the way over here, however long that takes, that's how long anaphase is. So usually it's the shortest phase of mitosis. Now I wanted to show a beginning and an end. So metaphase again had these chromosomes lined up down the middle, but now you notice that they're no longer X-shaped anymore. Half of the X is getting pulled to the right and half getting pulled to the left. It's almost like if you've ever gone fishing and you're reeling in your fish. So these just keep getting closer and closer and closer until eventually notice there's like no space between the centriole and the chromosome. It has been completely pulled over to the pole. So this would be one of the poles, this would be the other pole. So you still have a few maybe pieces of spindle fibers, you have your centriole, you have your chromosome, but you don't have sister chromatids anymore. Those have been pulled apart. So again, make sure you include both of these pictures in your notes. And this is just another way that anaphase can look, another picture that I found online. And it shows you the different chromosomes. So again, you notice that this is the same color as this to show that these are identical pieces, so that they've done that, so that you can see later when the cell splits in half that each cell is really identical to the other one. The last phase of PMAT is the T, and T stands for telophase. I've heard it pronounced telo or telo, so whichever you prefer. But telophase is the fourth phase of mitosis, and there are several things happening, and telophase really reminds me of kind of the opposite of prophase. So in prophase, the chromosomes became visible. In telophase, your chromosomes are going to unravel, so they're going to be chromatin. We also have the nuclear membrane is going to reform around your cluster of chromosomes, so they're not just floating around in cytoplasm anymore. They're going to be separated from everything else and be in a nucleus again. And in prophase, if you remember, the nuclear membrane was dissolving, was going away, so now the nuclear membrane is reforming. Your spindles break apart. We've already seen the spindles getting shorter and shorter as anaphase was pulling the chromosomes to opposite sides, but by the end of telophase, the spindles are gone. And the nucleolus, which disappeared in prophase, is now going to be visible again because we have a nucleus. So the nuclear membrane, the chromosomes, the nucleolus, all of that will be back to the way we're used to seeing it. Please make sure, again, you draw these pictures in your notes. You have room to do so. But at the beginning of telophase, notice that your nuclear membrane is very dashed. All right, that's because it's not fully grown yet. It's starting to reform. And notice you don't have the chromosome shape anymore. So chromatin is a stringy material. It's all of the information that you had wrapped up into a chromosome, only instead of having it all wrapped up, it is all like a single strand that's just floating about. So same genetic information, just it looks different. So we have the nuclear membrane, we have the chromosomes are unraveling. By the end of telophase, you have two nuclei. Notice you still have one cell. This is one cell membrane, but two nuclear membranes, two nucleoli, two sets of chromosomes. Everything is double. So all we have to do is cut our cell in half and we would have two cells. Our nucleus has already doubled. And that will take us to the end of telophase. I do have another picture here. Sometimes you'll see the cell membrane starting to divide. Notice that it's kind of pushing inward, and we'll talk about that in cytokinesis in the next video. Sometimes you'll see kind of like the pictures I had on the previous screen. Telophase is just the one cell, and it's still kind of its egg shape. It has not pushed inward yet. So depending on the type of picture you might see, both of these represent telophase. And again, it's because this is still one cell. We haven't cut this in half yet. Even though this is starting to push inward, it's not cut completely in half yet. So this is still all one cell in both of these pictures.